All right, thank you everyone for joining us and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Wednesday, May 28th, 2014. The topic for today's webinar is reactivating your immune system to fight cancer. Today's webinar is made, let's advance to the next slide, thank you. Today's webinar is made possible through the generous support of Peregrine Pharmaceuticals. Peregrine is a biopharmaceutical company with a portfolio of innovative monoclonal antibodies in clinical trials focused on the treatment and diagnosis of cancer. Peregrine is pursuing clinical programs in various cancers with their immunotherapy candidate, bavituximab, led by the Phase 3 Sunrise trial in second-line non-small cell lung cancer. Thank you, Peregrine, for your support. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute. Cancer Research Institute is the world's only nonprofit organization dedicated exclusively to harnessing the immune system's power to conquer all types of cancer. We fund scientists around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how cancer treatment today is undergoing a revolution thanks to these breakthroughs. After a 25-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your questions at any time throughout the webinar by typing in the Q&A box on your screen. Today's webinar is being recorded and will be available for viewing on the Cancer Research Institute website and on our YouTube channel within the week. Now it's my pleasure to welcome today's expert speaker. Dr. Rolf A. Brekken is an associate professor in the Department of Surgery at the University of Texas Southwestern Medical Center in Dallas. His laboratory studies tumor-host interactions with a particular emphasis on extracellular matrix and angiogenesis. His work is on the cutting edge of cancer immunotherapy research, and we are delighted that he could join us here today. As a reminder, we encourage you to type your questions into the Q&A box. And with that, I'd like to turn the presentation over to him. Dr. Brekken, welcome.
Thank you very much, Brian, for the introduction. And I just want to say at the outset, it's an honor and a privilege to have the opportunity to talk about some of the work that our group has been doing on a really exciting area, what we think a really exciting area on immune evasion. And so the title of my presentation is Reactivating Your Immune System to Fight Cancer. This could also be titled as a hallmark of cancer. Before we get started, I have a few relevant financial relationships to disclose. First, I consult for, I receive research support from, and I have stock or stock options in a variety of biopharmaceutical companies, including Peregrine Pharmaceuticals, who's sponsoring this webinar. What we're going to do today is talk about immune evasion as a hallmark for cancer and how immune suppression happens in the tumor microenvironment and some of the players that are critical to that immune evasion. And in particular, I'm going to highlight the function of a lipid called phosphatidylserine, or PS, in the immune suppressive environment of tumors. And finally, how antibody-mediated blockade of PS signaling can activate the immune system and lead to long-term durable responses. So my lab got into the immune therapy world by studying the tumor microenvironment. And what I'm showing you here are 6 hallmarks, and these were sort of put out by Dr. Hanahan and Weinberg in a review from about 2000. And these 6 hallmarks represent critical characteristics that tumor cells must, let me get this pointer here, these represent critical characteristics that tumor cells must acquire in order to be successful. So over the last 15 or 20 years, as you've heard through the webinars last summer, and you'll hear continually in the rest of the CRI webinars, let me move forward. Immune evasion is an emerging hallmark of cancer. And so a tumor, in order to be successful, must figure out how to avoid immune destruction. And so we're going to talk about how tumors do this by setting up an immunosuppressive environment and one of the key factors in driving that. So I think, first of all, one of the key points is to figure out who are the players in immune evasion and where are they in the tumor microenvironment. So I like this overview slide because it represents factors that are involved in anti-tumor immunity, which we'll talk about briefly now, and also factors that are involved in tumor-induced immune suppression. So if we think about an anti-tumor immune response, there are a variety of cells that need to participate in that anti-tumor response, and these include macrophages, such as M1 macrophages. And just parenthetically, very briefly, macrophages are immune cells that are present in all of our tissues and in tumors, and they can come in a variety of flavors. And we're going to categorize these anti-tumor macrophages as M1 macrophages. And another cell that's important to an anti-tumor immune response is a mature dendritic cell, which I'm trying to highlight here with the pointer. So mature dendritic cells are critical because they educate the adaptive arm of the immune response. And so we need to remember back to your basic biology where you learned that the immune system can have a memory. The immune cells can be educated to target specific antigens. And a critical cell in doing that education or providing that education to the adaptive immune response is a dendritic cell. But a dendritic cell must mature to be active. These mature dendritic cells interact with really the effector arms of the immune system, the adaptive immune system, and these include CD8 positive T cells or cytotoxic lymphocytes, shown here. Furthermore, there are other cells in the immune response, such as memory B cells, that are educated by these mature dendritic cells. So these are the cells that we want to recruit into tumors, and these are the cytokines listed over here that often drive a productive or anti-tumor immune response. And these include things like interferon gamma, TNF-alpha, and IL-12. Unfortunately, the vast majority of tumors that present already have tumor-induced immune suppression. And that's because they're populated by immunosuppressive cells. And we're going to talk in large part today about M2 macrophages and, let's see, I've just gone backwards here. I need a little help. Here we go. We have M2 macrophages, which drive immune suppression. We're going to talk a fair bit about these macrophages, as opposed to the M1 macrophages, which I brought up just a moment ago. And another cell type that we're going to talk a lot about are MDSCs, or myeloid-derived suppressor cells. And I want to characterize these myeloid-derived suppressor cells as sort of undecided cells. They haven't decided what they want to be when they grow up. So you can think of them as teenagers at a point where they can either decide to be productive for society or they can be, in essence, destructive. And MDSCs often sit in the tumor microenvironment, and they're in this undecided state, and they actually suppress the immune system. So these two cells, M2 macrophages and MDSCs, as well as a host of other immunoregulatory cells, drive immune suppression by secreting immunosuppressive cytokines, which are listed over here. Things including TGF-beta and IL-6, among others. So this is sort of a schematic of what the tumor microenvironment looks like, so an artist's rendition. There are lots of cells in the tumor. It's not just tumor cells as a bag of malignant cells growing out of control. In fact, these tumor cells, these cancer cells, recruit lots of host-derived or, quote, normal cells to participate in this thing we call a tumor. And the tumor I want you to think of more of as a tissue than anything else, with lots of different cell types. And so there are cancer-associated fibroblasts. There are endothelial cells which line blood vessels that help deliver oxygen and nutrients to growing tissues, including a tumor. There are support cells for these blood vessels called pericytes, and there are a host of immune cells, including these MDSCs and M2 macrophages. So this is sort of a schematic of what a tumor looks like. I thought it would be useful to show you what a tumor looks like in reality. And so this is a section of a prostate adenocarcinoma. I've taken it from a textbook. And what you're looking at here is called an H&E, a hematoxylin and eosin section. The dark purple areas, shown here that I'm circling, are actually the nests of adenocarcinoma tumor cells. These areas here are necrotic areas, and then the vast majority of the tissue, sort of this pinkish area, is really the stromal response. This stromal response is in large part the host cells that have been recruited convinced by these tumor cells to come in and participate in this thing we call a tumor. And so these fibroblasts, the stromal response is made up of extracellular matrix, fibroblasts, and lots of immune cells such as M2 macrophages and MDSCs. And this is where a lot of the action occurs with respect to immune suppression. I thought it would be useful to show you what these macrophages look like. And so I've taken a few examples from slides from other groups. And what they've done here is labeled macrophages. And in this first image on the left, macrophages are labeled in red, and tumor cells are labeled in green, and all the other cells are labeled in blue, right? And so the point of this is that there are lots of tumor cells, but there's also a tremendous number of macrophages that are in the tumor microenvironment, and they're spread throughout the entire tumor. They're interacting with tumor cells, they're interacting with host cells, other stromal components, and they're either being a pro-tumor macrophage or an anti-tumor macrophage. And unfortunately, in most cases, they're driving an immunosuppressive phenotype. If we look a little more closely at a tumor, and this happens to be an image from Johanna Joyce's lab at Memorial Sloan Kettering, here she's labeled macrophages in green, blood vessels in red, and tumor cells in blue. And the point I want to make in this image is that macrophages sit at the gateway where cells traffic in and out of tumors. And so they have a wonderful opportunity to influence immune cells coming in and tumor cells leaving. And this final image is a slightly different approach where they've labeled macrophages in green, but stromal cells in red. And so this entire field of view is actually a tumor, but we've only labeled stromal cells in red again and macrophages in green. And you'll see that these macrophages largely reside in these stromal highways that are supporting the actual tumor mass. So that's where these immune cells reside and can drive immune suppression in the actual tumor microenvironment. How does this work? Well, if we take, for example, MDSCs. I've told you that MDSCs are sort of undecided cells, immune cells, and they can drive immune suppression. So when we look at patient tumors and we see a high level of MDSCs, this correlates with a poor outcome in those patients. And this is in large part due to the fact that these MDSCs secrete immunosuppressive cytokines that prevent the development of tumoricidal M1 macrophages. They also prevent these dendritic cells from maturing. And so with an immature dendritic cell, it cannot educate T cells to drive an adaptive immune response. And thus you're left with naive T cells, which are ineffective at targeting or treating the tumor or attacking the tumor. In contrast, when you have a low level of MDSCs in a tumor, this correlates with a positive outcome in cancer patients. And this is in part due to the fact that there's typically a higher presence of immunostimulatory cytokines, which drive the acquisition of a tumoricidal M1 phenotype. So these cells are macrophages that can actually kill tumor cells. It also promotes the acquisition of a mature dendritic cell phenotype. And again, these mature dendritic cells actually educate productively T cells. And these activated, educated T cells can then go and seek out target positive cells. And these are often tumor cells. And thus, you can have an anti-tumor response mediated by your own immune system. So, what I've told you thus far is sort of just a primer, a very short, brief primer on immune evasion and how it's set up in the tumor microenvironment and the key function of particular immune cells in driving that immune suppression. What I want to spend the majority of the rest of the webinar on is a key pathway that participates in driving this immune suppression, and that is phosphatidylserine. So phosphatidylserine is exposed on the external surface of cells and vesicles in the tumor microenvironment. And this exposure is immunosuppressive. In fact, PS signaling is an immune checkpoint that's actually upstream of many of the other immune checkpoints that you've probably already heard of, such as CTLA4 and PD1 and PDL1. And finally, that antibody-mediated inhibition of PS results in immune activation and durable anti-tumor responses. So you're probably wondering, what is PS? Well, PS is a lipid, or phosphatidylserine is a lipid. Now, if you remember back to biology, you remember that your cells, all of your cells, are bounded by a bilayer of lipid. And that plasma membrane has two sides. There's the inner leaflet, which is shown here. So this is the inside of the cell. And there's an outer leaflet, shown here. And this is the outside of the cell. Interestingly, PS is segregated in normal cells to the inner leaflet. We don't find it in the outer leaflet under normal conditions. So looking at it another way, in this schematic, this is your plasma membrane, your bilayer, and PS is shown in red, and it's on the inner leaflet of the plasma membrane, and that's where it resides under normal conditions in normal cells. Now, PS can flip or be externalized under certain circumstances. One of these circumstances is normal programmed cell death. So cell death is a natural consequence of our body. All right, so cells naturally die, and when they do, they flip PS to the outer leaflet, and it serves as an eat me signal, and that's the actual term that immunologists use. It's an eat me signal. So this eat me signal can also be exposed under certain physiologic conditions, such as those found in the tumor microenvironment. These include oxidative stress, such as reactive oxygen species, hypoxia and reoxygenation, tumor cell metabolites, and cytokines. So in the tumor microenvironment, we have tumor cells that expose PS, there are endothelial cells that expose PS, and there's also microvesicles that expose PS. So why is this important and why do we care? Well, we care because externalization of PS leads to immunosuppression in the local microenvironment. So how does this work? Well, this is a rather busy schematic, but it helps illustrate how PS can drive immune suppression. So in essence, PS signaling suppresses immune surveillance. So when you have a PS positive cell, or when PS is exposed on the external leaflet of a tumor cell, or importantly, a cell undergoing programmed cell death under normal conditions, that PS is recognized by phagocytic cells, in this case, macrophages. And these macrophages, their job is to essentially clean up dead and dying cells. And if you think about it, you would like that to happen under normal circumstances in a quiet way. In essence, you can think of macrophages as garbage collectors. And under normal circumstances, the garbage man comes without waking up the neighborhood and alerting everyone, right? So PS engages a PS receptor on the phagocytic cell. Again, PS is an eat me signal. And this initiates a signaling cascade in these phagocytic cells to drive the expression of immunosuppressive factors, such as IL-10 and TGF-beta. In the presence of these factors, the immune system fails to develop a productive immune response. So you don't get M1 macrophages, which are or anti-tumor or pro-inflammatory. You don't get maturation of dendritic cells, and thus you don't get an adaptive immune response, and then you don't get T cells that drive target-specific killing. Why is this important? Well, it's important because PS signaling serves as a critical checkpoint for autoimmunity. So PS signaling has evolved to help us prevent autoimmunity. And in essence, tumor cells have hijacked this evolutionarily conserved pathway to drive immune suppression. This is a very early event. It's not the only event that drives immune suppression in tumors, but it's a very early event in the process of immune evasion. What happens when we interfere with this PS signaling? So we've developed PS targeting agents, and in particular we have an antibody called bavituximab that blocks PS immune suppression and activates a robust immune response. And there's lots of details in this slide, but I want to point out two things in particular. I told you in the previous slide that PS engages a PS receptor on the phagocytic cell, and that leads to the elaboration or the secretion of immunosuppressive cytokines. When the PS targeting agent is in place, what happens is it prevents that from occurring. So you block the engagement of PS with the PS receptor. This leads to the prevention of that signaling cascade in these phagocytic cells. So the other important feature of this PS targeting agent is that it provides a positive signal for these phagocytic cells. In fact, the phagocytic cell recognizes a portion of the antibody, in this case, bavituximab, or the PS targeting agent, and that portion is called the Fc portion of the antibody. It's recognized by specific receptors on the phagocytic cell, or the macrophage, and that is an Fc gamma receptor on that phagocytic cell. Engagement of that receptor leads to a different signaling cascade, which leads to the expression of immunostimulatory cytokines. So MDSCs can also recognize PS positive cells, and they can also be activated in the presence of a PS targeting agent by the Fc gamma receptor, the Fc mediated activation. And this leads to the differentiation of these undecided cells into mature dendritic cells and M1 macrophages. Furthermore, the PS targeting agent repolarizes these M2 macrophages into M1 macrophages. And so when you have M1 macrophages, these guys can be directly tumoricidal through a process called antibody-dependent cellular cytotoxicity, which we can talk about in the question and answer period. Furthermore, when you have mature dendritic cells, these guys are now capable of educating the adaptive immune response. And so T cells that they come in contact with are then educated to go target tumor cells, which express the appropriate antigen. And so you develop a long-term durable response mediated in large part by the activation of these mature dendritic cells. So, summary number one. PS is a global immune checkpoint that suppresses dendritic cell function, polarizes macrophages to an M2 phenotype, and induces the production of immunosuppressive cytokines while inhibiting the production of immunostimulatory cytokines, all of which promote T cell tolerance, and in essence, immune evasion. So if we look back at the hallmarks of cancer, there are strategies being developed by many labs across the world targeting each one of these hallmarks. There's 10 of them listed here. What we're going to talk about and what these webinars from CRI are really driving at are figuring out ways to block immune evasion. So this hallmark of avoiding immune destruction. You've heard about in the past strategies to block downstream immune checkpoints, such as CTLA-4, anti-CTLA-4, or anti-PD-1. And what I'm going to talk about for the remainder of the webinar is a PS targeting agent and the effect of this in preclinical models. So, in summary, tumors exploit multiple immunosuppressive mechanisms. There are downstream immune checkpoints such as CTLA-4 and PD-1, and we believe that PS represents an upstream and in fact a global immune checkpoint that influences many different features of the adoption of an immune evasive phenotype in tumors. So I'm going to give you a few examples and a little bit of data. This isn't going to be a data heavy webinar, but this is an example of an experiment done in an orthotopic model of lung cancer. And what I mean is that we've taken human lung cancer cells and we've implanted them. These are A549 cells that have been engineered so that we can see them because they give off light. We've implanted them into the lungs of rats. We did this at time 0. We then allowed these tumors to grow for about 3 weeks, and then we treated them. Now the therapy was either no treatment or control, radiation therapy, which is a common therapy for lung cancer, or the combination of radiation plus the PS targeting agent, in this case 2-AG4. So the green line is an image of tumor burden in the control-treated animals over time. And the blue line here, you can see that the treatments were given at 3 to 4 weeks, and there's a suppression of tumor burden, and then the tumors grow progressively. Whereas those that were treated with radiation and the PS targeting agent, you get a stabilization of disease and eventually a regression. In fact, half of the rats in this particular study showed no evidence of disease at 11 weeks post tumor cell implantation. So these are sort of gratuitous images at the 7th week or 4th week after therapy. So these are untreated animals, and the tumor is shown by this light image. You can see the red and the green and the blue. That's the tumor burden in the untreated animals. These are the animals that received radiation therapy alone, and these are the animals that received combination therapy of radiation plus a PS targeting agent. So this is at 7 weeks, or at this time point. If we look at 11 weeks or 7 weeks after therapy, you can see that there's very little tumor burden in these animals that received combination therapy. Another way to evaluate this is to look at survival. So this is an extension of this particular study, and these are still preliminary results, but we find them quite exciting. So it's the same setup, and what you're looking at is the survival of untreated animals, shown in the black line, animals treated with radiation therapy alone, shown in the red line, and animals treated with the PS targeting agent alone, shown in the blue line. So single agent therapy actually improved median overall survival. However, those animals receiving combination therapy, again, this is radiation therapy plus a PS targeting agent, showed remarkable results, such that at 180 days plus after implantation, we have 100% survival in this particular group. So we find this very exciting. And to support how we think this is working, I'm going to show you a few slides. So I've told you that macrophages are quite critical to either promoting immune suppression or immune stimulation. So we can look at macrophages and their phenotype by immunohistochemistry, and that's essentially what we've done here. We've taken tumors from animals that were treated with a control antibody or a PS targeting antibody, in this case 1 and 11, and we've evaluated the presence of M2 macrophages and M1 macrophages. And what you'll notice, and is quite clear, is that tumors from a control-treated animal have a lot of M2 macrophages, which is shown in this brown staining, but very few, if any, M1 macrophages. So this is the natural state of this particular tumor. However, when we block PS signaling with a PS targeting agent, we see the exact opposite. There are very few M2 positive macrophages, but lots of M1 macrophages, which we can see in this particular example. Additionally, if we look at the other cell type that we've talked about that drives immune suppression, in this case MDSCs, we can evaluate them in a very similar manner. So we can look at MDSCs by immunohistochemistry or by flow cytometry. What I'm showing you here is examples of tumors from a control-treated animal, and MDSCs for this particular example are yellow. So you can see lots of yellow cells in the tumors from the control-treated animal, whereas there are very few yellow cells in the tumors from animals treated with a PS targeting agent, in this case 2-AG4. And this is quantitated here. Furthermore, we get similar results when we use flow cytometry to analyze the tumor, which is a different way of looking at the cellular content of a tumor. So when we change the macrophage phenotype, or we drive an acquisition of an M1 phenotype, and we prevent the accumulation of MDSCs in the tumor microenvironment, if what I'm telling you is correct, we ought to have we ought to have a durable long-term response in tumors. And so this was illustrated in a study published by the Thorpe Lab a few years ago, in which they took a model of glioma in rats. And they implanted these cells into the into the brain of these animals, and then they treated these animals with a control, which is shown in the black line, or radiation, or PS targeting agents. So that single agent therapy are shown in the green and the blue line, and you can see that they improved overall survival. However, when they did the combination of radiation therapy, one dose of high dose radiation, plus the PS targeting agent, we see a substantial improvement in survival. And we had about a 15% of animals that showed long-term durable responses. Now, they did something quite intuitive at this point. They then re-challenged these animals that were showing a long-term durable response with the same tumor cells. And what they found is that all of these animals were now immune to further implantation with those same tumor cells, strongly suggesting that they had developed an adaptive immune response with memory. So to prove this, they took T cells from these animals and they evaluated their ability to target tumor cells in vitro. And what they found is that T cells harvested from animals treated with a PS targeting agent showed specific tumor cell cytotoxicity, whereas those T cells harvested from a control-treated animal that had a tumor did not show any specific in vitro cytotoxicity towards tumor cells. We have a similar example in a genetic model of prostate cancer. So these are mice that spontaneously develop prostate tumors. They're called tramp mice. Now, prostate cancer is typically treated with androgen deprivation therapy, and we've modeled that by evaluating the effect of castration in this particular setup. So animals treated with a control antibody are shown in the black line. And I should say that this is a survival curve. So this is the median survival of tramp mice. And when you treat them with castration, shown in the red line, you get an improvement in survival. However, when you treat them with a PS targeting agent, you also get an improvement in survival. But when you treat them with both castration or androgen deprivation therapy plus the PS targeting agent, you get a greatly enhanced improvement in survival, and you further get a long-term durable response. And what I'd like to point out is in this genetic model of prostate cancer in mice, tumors are constantly being developed. Tumors are constantly coming up in the prostate gland. However, these animals, these 40% of the animals that show a long-term durable response, have an immune system that's now capable of removing those tumor cells. So this is a summary of what I've told you thus far. And that is that under normal circumstances in a tumor, PS drives the acquisition of an immunosuppressive phenotype. And in fact, MDSCs and M2 macrophages engage PS via a PS receptor, and this leads to the expression of immunosuppressive cytokines, which facilitates the recruitment of M2 macrophages and prevents the development of mature dendritic cells, and thus you're left with naive T cells that now tolerate the tumor. When we interrupt this signaling cascade with a PS targeting agent, we not only block PS signaling, but we provide a productive immune signal, the FC portion of the antibody, that drives the acquisition of an immune stimulatory phenotype. And this leads to the expression of immunostimulatory cytokines, the development of mature dendritic cells, the acquisition of tumoricidal M1 macrophage phenotype, and these macrophages can then use ADCC to kill tumor cells specifically. Furthermore, mature dendritic cells educate the adaptive arm of the immune system, so you get activated T cells that then show tumor-specific cytotoxicity. So in essence, blocking PS drives MDSC differentiation and dendritic cell maturation. So if you've listened to a few of these webinars, or you've been following the immune therapy field, you're probably asking the question, what happens if we combine an upstream immune checkpoint inhibitor, such as a PS targeting agent like bavituximab, with a downstream immune checkpoint blockade, such as anti-PD-1 or anti-CTLA-4? So these are strategies or experiments that the laboratory is now pursuing. I'm going to show you some very preliminary but exciting data on those very things. And so this is an example of combining a PS targeting agent, in this case mouse chimeric 1N11, with anti-PD-1 in a syngeneic model of mouse melanoma. And I want to just point out this particular graph here. You're looking at the effect of PD-1 therapy alone, shown in the blue line, and it does show some efficacy, but there's a spread in tumor volumes over time. However, when you add the PS targeting agent to the anti-PD-1 therapy, you get a much greater tumor control, and it's a much tighter distribution of tumor volumes over time. Furthermore, you drive a productive immune response, which we can then evaluate through other measures in these animals. A similar story when we look at another prominent immune checkpoint blockade, and that being CTLA-4. So when we take CTLA-4 alone and we treat animals bearing melanoma, we have some measure of tumor control. However, when we combine CTLA-4 with a PS blocking agent, we have a greatly enhanced response in terms of tumor control. And so these studies, these preclinical studies, are highlighting the fact that you might be able to combine different immune therapies, especially an upstream immune checkpoint inhibitor with a downstream immune checkpoint inhibitor, to have a greatly enhanced effect. So, key messages are what I've tried to get across in this relatively short webinar. One is that phosphatidylserine, or PS, is externalized in the tumor microenvironment and is a major immunosuppressive signal. In essence, we think PS is a global immune checkpoint, and that antibody-mediated blockade of PS signaling can break immune tolerance by reactivating both the innate and adaptive immune system and result in durable anti-tumor responses in multiple preclinical models. So combination studies with, preclinical combination studies with other immune checkpoint inhibitors are currently underway and looking quite promising, and early phase clinical studies are also planned. So I'd like to make a few acknowledgments. First, all of this work was done by the Phil Thorpe's lab at UT Southwestern and my lab at UT Southwestern in combination or collaboration with our biopharmaceutical collaborators, Peregrine. I like to say that my Ph.D. mentor, Phil Thorpe, is the creative force behind this work, and it was really his life's ambitions to see this strategy move forward, and he really was the one who was driving this project. Phil, unfortunately, passed away about a year ago, and it's now my pleasure to have the opportunity to help drive this further. So with that, I'd like to close the presentation part of the webinar and then open it up for any questions.
Dr. Brekken, thank you so much for that very interesting presentation. We did get quite a number of questions from the audience. Some of them are similar to one another, so I'll start with those. So the first one comes from Andrew, and he asks, if tumor cells express the PS eat me signal, why don't the macrophages just eat it?
Excellent question. In essence, Andrew, a lot of tumor cells are subject to what we call phagocytosis. But the critical thing is that when those phagocytic cells, when the garbage collectors are actually eating those tumor cells, they're also secreting immunosuppressive signals that prevent the recruitment of immune cells that might have a more productive anti-tumor response.
Okay. So then Ron asks, why does the eat me signal change when cancer expresses PS? Is there a specific threshold between eat me and I'm not quite dead yet?
So I guess in the context of my presentation, I really didn't get into the specifics of the eat me signal. But tumor cells, I think, not all tumor cells will express PS. And one of the things that I failed to highlight is that those cells that express low levels of PS really drive that immune suppression phenotype. But tumor cells, we know, are proliferating rapidly, much more rapidly than other cells in the tumor microenvironment. So they're repopulating. The other thing to remember is that when we treat with standard therapies, we're driving tumor cell death And while we're going to kill some tumor cells, we're also driving this immunosuppressive pathway. And so that's one of the reasons why combination therapy seems to be quite effective in this setup, in this strategy. So I guess I didn't answer that question completely, but there does seem to be a threshold for how much PS is exposed and whether you're actually getting phagocytosis as well as immune suppression or just immune suppression. Maybe that was a shorter answer.
There was some question, and I think you did touch on this in your talk, but perhaps you can just restate it or clarify. Does the PS cause M2 cells to convert to M1 cells, or is the PS causing the recruitment of M1 cells?
So PS, the presence of PS in the local microenvironment serves to drive immune suppression. So it's recognized by phagocytic cells, which then leads to the expression of immunosuppressive signals that prevent the recruitment of productive immune cells or anti-tumor immune cells. And what happens when we have the PS targeting agent is you flip that. In fact, in vitro, we can take M2 macrophages and treat them with the PS targeting agent and differentiate them or repolarize them to M1 macrophages. Does that help clarify that to some degree?
So there is some direct effect on the biology of the cell. It's not just a matter of recruitment. There is some form of transformation happening at the cellular level.
Right. So we think that signaling from the PS receptor is quite important, and I left that kind of flapping in the wind on purpose because there are many different cell surface proteins that can serve as a PS receptor. And one of the things the lab and many labs across the nation are looking at is which PS receptors are most relevant for driving this immunosuppressive signal.
Okay. This was an interesting question, and I think this is probably right up your alley given your focus on the stromal tissues around the tumor and the tumor microenvironment in general. So the question from Rashinda is it just tumor cells that show or express PS on their surface, or do other cells in the tumor microenvironment express this, like stromal cells or fibroblasts?
Right. So this is an excellent question, and it kind of leads into how we discovered this. Well, we didn't discover it, but how we recognized this pathway. PS is externalized on many different cell types in the tumor microenvironment, and any of those cell types can drive this PS receptor-mediated immune suppression by phagocytic cells. So we came about this from the vascular angle. So we initially developed these PS targeting agents as agents that can specifically target tumor vasculature. And what we found is that while they do target the tumor vasculature, they also have this rather potent immune stimulatory phenotype.
Okay. Another question, and we're almost out of time, but I think we have just two more, I think, brief questions. What is the expression pattern of PS on tumor cells? Meaning, is it homogeneously expressed on all the cells within a tumor? Is it only expressed on some of the cells? And
right. So I'll try to answer that.
Is it associated with the grade or stage of the tumor?
Right. So this has only been evaluated preclinically because, as one can imagine, it's difficult to look at PS because you can't just stain tumors with an anti-PS antibody because PS is actually in all of our cells. What you need to find is PS that's externalized onto the outer leaflet of the cell. So we've looked at this in a wide variety of preclinical models, and the range of exposure of PS is quite wide in terms of how much PS is present. So it's not homogeneous, and it's not because if you're a tumor cell, you don't automatically express PS on the outer leaflet. However, when you put cells under therapy, tumors under therapy, you universally upregulate the externalization of PS in the tumor microenvironment.
All right, thanks. And we have a question from Matt. Are there any drugs beside bavituximab that target PS, and if so, are you aware of what stage of development they might be in?
Okay. I'll go back to the other question because I didn't answer grade or stage. At this point, it's not clear whether PS exposure has any influence on grade or stage in clinical samples, and in large part because of the rationale of why that is because of what I just told you. So in terms of other strategies to block this pathway, there are actually a number of other strategies to interfere with this cascade, and they often center on agents or drugs that block PS receptor signaling directly. And so there are a variety of companies that have agents that interfere with other specific PS receptors, such as the TAM receptors or Tyro, Axel, and MER. There are antibodies or agents to interfere with TIMs, TIM3 and TIM4, and they are in the preclinical stage of development for the most part.
All right, well, thank you very much. That's all the time we have today for questions. Dr. Brekken, thank you again so much for joining us. Excellent presentation and very interesting talk. Another angle to the global efforts to mobilize the immune system to fight cancer. So before we conclude, I would like to thank once again our generous sponsor, Peregrine Pharmaceuticals, whose generous support has made today's webinar possible. I'd also like to notify you of our upcoming webinars. All of these, including Dr. Brekken's, I forgot to mention at the beginning, are being rolled out as part of our Cancer Immunotherapy month, which is June. So throughout the month of June, we will show an additional 4 webinars, all touching on different subjects on how cancer immunotherapy is helping to revolutionize treatment today. So these are these. You can register at cancerresearch.org/webinars. Also, if you are interested in learning more about Cancer Immunotherapy Month, we have a range of activities throughout the month of June in which you can participate and add your voice to the movement. That's at cancerresearch.org/join-the-movement. And lastly, for those of you who are interested in finding clinical trials, either of bavituximab or any of the other immunotherapies now in development, the Cancer Research Institute provides a clinical trial finder service on its website, and that's cancerresearch.org/clinical trial finder. That's all for today. Thank you all very much, and I wish you all well.